Hi everyone. Okay, so we are going to dive in today. I hope you enjoyed the dog video. So I have to say, if you've ever heard me when I've been recording at the wrong time, you probably heard some barking in the background. So I've always been fascinated by the evolution of dogs, and knowing that we've probably been together for at least 30,000 years actually makes it pretty interesting. So the cool part and the role that domesticated dogs and animals played with regards to Darwin is that Darwin noticed that we had selected for and against certain traits in these critters that were domesticated. And he felt that there was no reason those rules wouldn't work under nature too. Okay, so they definitely played a role, and he was thinking about in particular dogs and pigeons and corn and so forth, and that helped him shape the concept of evolution and natural selection. Just to give you an idea of the lineup for Friday, what we're going to do is we're going to have one lecture, okay, because I don't want to heap two lectures on you and then have to have a whole bunch of new information to know before the exam on Monday, okay? And then I will probably over the weekend randomly throw some practice questions out at you for you guys to try, all right? Then what will happen is Monday there's no lecture, because I want you guys to focus on the test. The test will be released at 2 o'clock on Monday, and you'll have until 2 o'clock Wednesday before class to turn it in, all right? And so you guys, from what I heard, you rocked the discussions, did really well going through the questions. And so I'm very happy because that means I'm doing my job well. So when you guys do well, then it makes me very happy because otherwise I'm doing something wrong. So yay, nicely done, everyone. So we'll talk about Darwin and Wallace's big ideas, which is basically natural selection. We'll talk about reading phylogenetic trees and how to build one and all sorts of other good stuff. And I hope you guys can see this, this figure because it's hilarious, and I'm just shaking my head at the figure at the bottom right. Okay, so yeah, that's a disaster waiting to happen. Please don't ever do that. So in the 1700s and 1800s, there was a strong push by the church, which back in the day the church was very powerful. And you basically did not defy the church. And so the church was, very influential to the government. And most people back then believed that species were fixed entities and that they did not change. Okay. Well, along would come two scientists, Darwin and a man named Alfred Russel Wallace, who basically both kind of discovered natural selection. Now, I don't know if you all know the story behind natural selection, how it was discovered. Darwin's grandfather, actually, Charles Darwin's grandfather, whose name was Erasmus Darwin, actually wrote about changing with modification. So he kind of grew up with these ideas, and they were in his family, and he was groomed to think along those lines. And we'll talk a little bit later more about the details of how he discovered it. Darwin tends to get most of the credit by natural selection. And the reality was, though, is another man named Alfred Russel Wallace was kind of publishing a paper along the same time as Darwin. And technically, they both discovered it together, although Darwin definitely receives most of the credit. For some strange reason, it just kind of happened that way in history. So what Darwin and Wallace both independently realized was that you could find fossils of extinct animals found where animals that look similar live today. Okay. They also realized that islands near each other have similar but yet distinct animals living on them. The finches are a great example of that, and in particular that was influential for Darwin. Some species would also possess vestigial structures like finger bones and whale flippers and so forth. In other words, structures that are no longer needed, but maybe at one time was what they were hypothesizing. Also, that individuals within a given species could vary in small ways. We know that. Okay. And then animal populations are kept in check because many of them die when they're young, okay, in every generation. And that came about through a thought by Thomas Malthus, who wrote the essay Principle on Population, who felt that there was a struggle for existence. And this was in humans back in the day as well as in nature. So the way the publications went is that Darwin went on this journey where he traveled the world, and Alfred Russel Wallace also did the same. And then Darwin would collect enough information to eventually write his Origin of Species that would be published. And Wallace would write a paper on mites that was very similar that also proposed some of these very ideas. As Wallace was traveling the world, one thing he noticed is that there was a group of islands in the archipelago, okay, that were very close to one another from a geographic perspective, but had some very different animals on them. So, and eventually came up with what would be called the Wallace line, where he would draw this division. And critters that were found to the west of that were placental mammals, okay, whereas those that were found to the right are marsupial mammals. And his question was, how in the world could you have islands that are so close together that have such a different composition of animals? Well, eventually, asking these questions would lead him to be the father of a field of science called biogeography. Write that down, biogeography, okay? In which case, he was interested in how animals and plants and so forth came to live in the areas that they do. Eventually, it would be found that this is due to a process called plate tectonics, whereby we know now that the plates move around and so forth. And that's what happened is they took the animals with them. Okay, but remember, we take all this knowledge for granted today, but back in the day, we didn't know this. Now, a little curious fact about Alfred Russel Wallace is that he was a self-made man, okay? And so he earned his living by traveling the world and collecting samples and bringing them back for people. Now, another little known story is that he spent years doing this, okay? And then, unfortunately, one day, the ship that he was on would run into trouble. And in fact, with all of his samples, the ship would sink. And can you imagine collecting samples, traveling the world for years and years and years, and all of a sudden your samples sink? The crew died. He's the only one that made it. Okay. And then he did make it back home. Fortunately, he was smart enough to insure everything, so he didn't take a total loss. But I imagine his heart was breaking as he saw all those samples that he was bringing back, in addition to the crew going down. How horrible. Now, one of the things that Darwin noticed as he sailed around the world, and by the way, he sailed around the world on a ship known as the Beagle. Now, a very little known fact. When I grew up, Darwin was sort of villainized in the educational system, because this has been a really long time ago, as my daughter likes to remind me. And so Darwin was, yeah, he was made to be an evil person. Well, it's really sad that that happened, because in reality, Darwin was a very kind person, according to anybody who knew him, and a very gentle man. And the idea was, though, and why everybody was having a fit, is because he was challenging the church back in a day when you did not. Okay. Now, Darwin did travel the world on the HMS Beagle. His father actually almost didn't let him go on that trip. Can you imagine how different science would be? He was originally training to be a country parson and running his own church. So another little fun fact is Darwin's father. Now, Darwin came from a wealthy family, which differs from Alfred Russel Wallace. And Darwin's father initially wanted him to be a doctor. Well, this was back in the 1800s, back before anesthesia. I don't know if y'all can imagine this, but Darwin went to observe his first surgery, which was an amputation with no anesthetic for the patient. Okay, so imagine that you're going in and you're watching the surgery for the first time. This poor patient has no anesthetic and he's supposed to have a leg amputated. There was blood, there was screaming. Darwin couldn't handle it. I wouldn't have been able to handle it, and he turned around and ran out. Well, his father just kept saying, okay, so you don't want to be a doctor, why don't you be a country parson? You can go to school for that. And he agreed to do so. So he went to college, training to be a country parson. Well, then he heard about this ship, the Beagle, that was going to sail around the world. And he decided he really wanted to go exploring. Well, his dad wasn't really all that thrilled about it at first, because he was like, come on, hurry up, get a job, figure this out. Not going to lie, I've had the same conversation with my daughter a time or two. However, his uncle talked his father into not only letting him go on the trip, but financing part of it to help Darwin find himself. And so that's how Darwin ended up on the Beagle, going all around the world and visiting the Galapagos Islands. Well, when he got back, he knew that, creatures seemed to change. And he started to get these ideas that he was a little nervous to publish right away, and he was pretty meticulous. And so he collected his data, and he, analyzed it and reanalyzed it and reanalyzed it. And that's when he finally started to come up with the idea for the origin of species. Well, he had a lot of friends back in the day that he was talking to about this idea. Now, something that's a little sketchy, once again, I'm always saying we should publish a book on, the seedy side of science or something like that. Because his friend Charles Lyell, after about 20 years of Darwin sitting on his data, okay, and thinking about it, Charles Lyell received a paper from a man named Alfred Russel Wallace on this idea of natural selection. Well, Alfred Russel Wallace went back to Darwin and said, hey, you better hurry up and get this done, because otherwise someone else is going to publish your idea. Now remember, they came up with it independently. Today, by the way, you cannot do that. You are supposed to review papers, keep your mouth shut, and never say a word to anybody, and whoever publishes first, publishes first. So that's where things get a little bit sketchy. What ended up being done in the end was that they were to both present this idea at a conference together. And so then they were both supposed to receive credit. Well, Alfred Russel Wallace still doesn't receive a whole lot of credit for it, even though, his idea was revealed at the same time as Darwin's. And sometimes things like that just happen. But either way, they both observed that certain species were, similar and found in distributions and so forth. And they both came up with the ideas technically. So really, they both should get the credit. But, we do the best we can to tell what happened and represent things, as they stand. Okay, so in class I try to present questions in all sorts of different manners because I want you guys to get used to these because despite the fact that our exams are not multiple choice questions, if you all choose to take 172 and so forth, you're going to see those, okay? So what I want you to do is to take a moment, read through this, okay? Pause lecture right now, and then decide which information you feel is correct, okay? So hit pause, and then we'll come back and finish. Okay, so as you go through this, okay, and as you're looking through the different possibilities, all right, so what you want to do is you want to read through all the different choices. Okay, so choice A, well, organisms on islands evolve faster than those in the mainland. Well, there really isn't any evidence for that, and they really didn't say that because it depends on the critter and so forth. So I would cross out A. B, size, shape, and many characteristics have changed over time. Ah, that one sounds pretty good. Okay, keep reading. C, geologic processes are not involved in the evolution of organisms. Well, no one ever said anything about that. Okay, cross that one out too. And then D, a wide variety of unique species can come from a common ancestor. Ooh, that one sounds pretty good too. Therefore, when you're looking through this, go for E. Okay, so unlike as suggested by the church, creatures will mutate and they will have descent with modification, which is the whole premise of natural selection. All right, hopefully that makes sense. Okay, so as we're going through and looking all the different, reading all through going through the different topics, goodness, we're now going to focus on some species possess vestigial structures. Okay, so this part's actually pretty cool. So what we're going to do in the next slide is we're going to look at a whole bunch of distantly related species, okay, and we'll see that they actually have quite a few characteristics that are similar that you wouldn't necessarily expect. Okay, so the term for descent with modification, as it applies to a specific trait, means homologous. Okay, so homology means you have similarities due to common descent from a shared ancestor. All right, so that's a homologous trait. Now, if you look at the appendage of turtles, humans, horses, birds, bats, and seals, these guys are all distantly related. But the cool part about this is if you look at them, they all have a humerus, they all have a radius and ulna, they all have carpals, they all have metacarpals, and phalanges. Okay, so the reason they have all these is because a common ancestor had them and then passed them down, to them. Now, they've been modified over time, and that's fine. But when a common ancestor had these traits and then all the descendants have these traits, that's called a homologous trait. All right, so homology versus similarity due to a common ancestry. Forelimb bones happen this way. DNA sequences happen this way. The proteins, 20 amino acids happen this way. ATP, membranes, all of that. All of these traits are because there was, we all share them with a common ancestor. We have these too, obviously. Now, another term you need to be familiar with is called analogy, or an analogous trait. This is when you have a trait that evolved independently in different organisms because they adapted to a similar environment. Okay, a great example of this is the, hydrodynamic shape of dolphins and fish. Okay, they both look the same. However, it's not because their common ancestor necessarily had that shape. It's because of the fact that it was called convergent evolution, by the way, is another term for this. Write that down. Convergent evolution. Okay. Bat wings, bird wings, and butterfly wings. All right, so insect wings are actually very different than mammal wings, like bats and birds. However, there's a serious advantage to being able to fly through the sky. You can escape predators and so forth. And so it's not because a common ancestor had them, though. Okay, so it was convergent evolution. Additionally, sweet and regular potatoes, just the way they evolved. Webbed feet in frogs and ducks. not because of a common ancestor, but because of how, they converge in evolution, as they evolved over time. All right, so the next thing we're going to focus on how individuals within a given species vary in small ways. And all you have to do, by the way, is look at humans. Look at all the cool different variation we have. That's what makes it so much fun, okay, is that we're all different. Nobody's the same except maybe identical twins. And I'm not saying that's not cool, don't get me wrong. However, just humans in general, they all have variation. Every species has got variation within it, and you have to have that in order for them to be able to evolve. It's essential, okay, for the long-term survival of a species. Now, a great example of this is this small little edible saltwater clam. I have to tell you, these are one of my favorite examples because when I was a child, I remember going on the beach with my grandparents, and they would let me collect them in a bucket and bring them back to where we were staying in the hotel, and I always had such a great time with them. Fortunately, they also taught me to let them go when we were done, but they were still actually pretty cool. And so there's a wide range of possible colors. They can be almost white through pink, orange, red, and purple to brownish. They can have rays. They can have light rays and dark rays. They're all very cool. They're all the same species, okay, but you can really see the variation, which makes them pretty awesome. Plus, one of my favorites. Now, another idea that shaped Darwin was written by a man named Thomas Malthus, where he wrote on the Essay of Principles of Population. And sadly, he was writing about humans, which kind of is depressing. And he was talking about there was a struggle for existence and more offspring would be born than can survive. Now, unfortunately, he was right, and it still makes me sad. However, this is very much the same way in nature. Okay, so populations were kept in check because of the fact that there will be a struggle of existence. And, that's just the way it is because there's not enough resources for everybody. Again, sad, but that is part of nature. And, it doesn't. Hopefully things, keep getting better. Okay, so what I want you to do once again, applying and getting you used to taking multiple choice tests, okay, because you're going to have to do it in 172. I want you to stop, to read through this, take a moment, and come up with the correct answer, okay? Go ahead and pause and read through it, and then choose your best one, and then start us up again. Okay, so if we're looking through this, okay, so individuals within a species vary, populations are kept in check. What did this lead Darwin and Wallace to infer? Well, only the strongest and fastest individuals will be able to reproduce. Well, okay, when you're reading through these, in order for you to choose it, it has to be 100% correct, okay? Unless specified otherwise. Sometimes they say best fit, and I hate that because I was like, but I'll never do that to you. But just realize it's in the details, right? We've said that before. So A is not the case, though. Okay, because only the strongest and fastest, no, that's not the case. So cross out A, which also, by the way, eliminates D. Okay, so for B, smaller and slower organisms will be selected against. Well, that depends, because it depends on the scenario. There are lots of times when being smaller is actually a benefit. Okay, so cross out B. Then look for C. Okay, individuals whose inherited traits gives them a higher probability of surviving and reproducing in a given environment will have more offspring than other individuals. Aha, this is the correct answer. Okay, so again, always read thoroughly, okay, and make sure you've read all the wordings to know that you've got all the information you need. Okay, so we know that in nature, organisms produce far more offspring than can survive. Now, the best example of this I can think is as a maple tree. So we have a maple tree in my backyard, my husband and I do, and oh my goodness, all the offspring that start growing in that maple tree. And so more of them are produced than can survive. But this is what led Darwin and Wallace to focus on differential survival and how that plays a role in evolution. Okay, so differential reproduction we know now is the most important part. So those that have the better traits and can be more successful will have more offspring, okay? And so that tends to be more important from an evolutionary perspective, but that took years to discover, okay? So both of these things are very, very true. Okay, so Darwin and Wallace came up with two big ideas, and the first one is called descent with modification. So life on Earth had a single origin, and all the diverse critters we see today are descendants and modified by evolutionary processes of this common ancestor. So remember, this directly contrasted with what the church was suggesting. Now, I'm not touching religion with a 10-foot pole, okay? And the one thing I'll say is that what we teach in here is based on science and the scientific method. And the reality is, in my own personal feelings, is that science and religion are two completely different things. So religion is based on faith, and we don't even have really a way to test for that one way or the other, okay? Science is based on the scientific method and testing hypotheses and so forth. And so the reality is the two are kind of like apples and oranges. Personally, if I live long enough, I'm going to go back to school. I'm going to get another degree with a PhD in religion because I think religion is actually fascinating. And seriously, how cool would that be? PhD in religion and PhD in evolution. That would be so cool. Yeah, one of my life's goals. Okay. But what we will stick with in this course and the way we will treat this is that we will focus on what we can talk about with the scientific method, okay, testing hypotheses and all that other good stuff. And, we'll leave everything else behind. The other idea that Darwin and Wallace came up with was that natural selection would explain evolution. Okay, so those that have heritable traits give them an advantage in a different environment, so they produce more offspring than those without the traits, and therefore those alleles are favored, okay, as an increase in frequency from one generation to the next. Now, I want you to write this down. In order to define evolution, it is changing allele frequencies through time, okay? Changing allele frequencies through time. I was taught by some of the best evolutionary biologists, and I am very grateful for that. And that was always how I was taught to define evolution, changing gene frequencies through time. Okay, nothing more complex, nothing more simple. I think that works out perfectly. So now we're going to work through an example of natural selection, and then what I want you to do is write down what are the conditions necessary for natural selection. Okay, you have a population, for example, of bugs. Okay, and there's multiple colors. Well, the birds come in and they naturally go after the ones they can see the best. Okay, so who's left over? The ones that are better camouflaged. And given that information, your population changes because those colors are based on alleles. Okay, so you start out with a population at the top of both green and brown alleles, and you end up with a population at the end of only brown alleles. Oh, look, changing gene frequencies through time. Okay, so what are the conditions that are necessary for natural selection? Stop the slide, think about it, and then write it down. Okay, so in this example, you have to have variation in traits, okay? In this particular example, they have the carapace, which is the outside color of the bugs, okay? Then that trait has to be heritable. So heritable means it gets passed on from one generation to the next. Then you have to have differential reproduction. So tan beetles reproduce more than the green beetles, and of course they're the ones that survive more. Therefore, their population evolves. Okay, in this particular example, we went from a mixed population of green and brown to only brown. Again, changing gene frequencies through time. So what happens is ultimately your populations will adapt, okay, where they'll have heritable traits that increase the survival or the reproductive status of that creature. For example, if this was me, I totally would go after that and be eaten by the shark in 2 seconds, okay? Big fan of ice cream, gotta tell you. So, but just realize that when things, help a species do better or help individuals do better, then they adapt to their environment through adaptations. Now, I am old enough, as my daughter likes to remind me, I'm old, that I remember when National Geographic actually came out with this edition. Okay, and the reason that they came out with this, by the way, was because even now, the whole concept of evolution is one of the most controversial fields of science. And the reality is, though, you read the National Geographic heading and then you open it up, and the fact of the matter is the evidence for evolution is overwhelming. It's in every single field of science, from biochemistry to biology to microbiology to it's everywhere. Okay, the reality is evolution is overwhelming. The evidence is overwhelming. The reason it's so controversial, of course, is because it challenges the church. And so again, that's based on faith and it's apples and oranges. And the reality is all we can do here is to talk about the scientific method and what you can show with the scientific method. Now, personally, I think both of them can live peacefully, okay, because they're both after the truth. And I will tell you, I've seen evolutionary biologists that attend church every Sunday. Okay, I've seen others that don't believe in a higher power at all, and that's totally fine. Everybody's allowed to believe what they want to believe, but again, I think they can both exist peacefully. I just believe that religion should be taught in religion classes, and then science should be taught in science classes based on the scientific evidence, and that's the way most universities tend to treat it. And I think as long as you do that, everybody's good. Okay, so now we're going to talk about phylogenetic trees. So the relationship of critters to one another are represented in trees. And so what we're going to focus on now is how to read a tree and how to build your own tree and the different process of speciation, all sorts of other cool stuff. So one thing I will tell you, when I was an undergrad, they didn't even offer an evolution class, evolutionary biology class at my college, which was surprising because, okay, it wasn't that long ago I was in college. It wasn't until I got to grad school that I realized this entire field existed and I realized how fun it is and how much it explains the natural world. So I didn't even get exposed to phylogenetic trees until grad school, which, whoa, took a while. Okay, so the way that you read these trees is when you see a node, which is at the bottom center, that's a speciation event. Okay, and before that is the ancestral population, then you have a node, then you have the descendant populations, then you have more nodes, then you have more descendant populations. The way that time is represented is time is at the bottom, past, okay, and then the more recent is towards the top. Now, hopefully, you've done some reading, like was assigned, okay? And you should be able to define a phylogenetic tree and a phylogeny and think of some key ideas and how to interpret this, okay? So stop again, take a moment, fill this out, and then start up lecture again. By the way, the reason that I try to have you do so many in-class things, even though we're being taught remotely, is that research shows if you sit there and you just listen to me ramble, it's not going to stick. Okay, so the reason I have you stop lecture, that I ask you to do so, and then think through ideas yourself, is because if you think through it yourself, whether you're right or wrong, you're much more likely to remember the idea, especially if if it's similar to what I'm thinking, okay? So please do take the time to stop and make this an active thing where you actually think through, okay, and then continue once you've put all your ideas on paper. So in this situation, the key idea, closely related species share many characteristics, and that should make sense. Closer related are, the more traits they show. Distantly related will share fewer characteristics, okay? A phylogenetic tree is just a graphical representation of the evolutionary history, basically who's related to who. And a phylogeny is a hypothesis about the evolutionary history, okay? And the reason it's a hypothesis is because as we learn more, trees get rearranged and people, change ideas and so forth. And it's actually pretty cool. And that's part of the fun of science, is learning how things, really worked back then. And sometimes there's a lot of, disagreements, and that's okay. As long as you, are trying to figure out what really happened, it's all information. Okay, so let's talk about the process of speciation. So speciation is when you have one species that turns into multiple species. And in order for this to happen, it begins with, having gene flow stop between two different populations for some reason. Okay, in the example here, you get a large population, and then all of a sudden you get a mountain range, and those two populations are separated, and they're no longer exchanging genes. And that's key, by the way, in order for them to actually split into two different species. Now, the idea is over time, your separated populations will accumulate new mutations, okay? And so they can accumulate so many mutations that even if they were allowed to get back together, they would not be able to interbreed, either genetically or behaviorally, or there's going to be some barrier that keeps them from interbreeding. And once they're no longer able to interbreed, then, according to the biological species concept, they are two distinct species. Okay, how we define species is different over time, and we'll talk more about that later in the term. The most popular way to define species is the biological species concept. You should write that down, okay? And it just bases species on, you're considered a separate species if you can no longer produce fertile offspring. Okay, so keep that in mind as we go through lectures. Okay, so very often what happens is that the tree is not as simple as the way we've been representing it, okay? So if you actually have a tree of different generations of, for example, insects, okay, and you're looking at, the insects and how they've changed and had different lineages over time, we're going to look at one particular example where we have an insect here named Mimi, okay? And so Mimi has siblings, and she also has parents, all right? And so if you look over, you can see that on the upper left. Look at the upper right, that's where Mimi actually fits with her siblings and her parents and so forth. And you can see, especially with insects, they'll have a lot of offspring, a lot of critters over time, and especially if you're dealing with generations, from the 90s through the 2000s. And I know all of you probably don't remember those days. Sadly, I do. So let's take our data and actually apply it. Now, in the reading that you've already done for today, ahead of time, okay, you should have enough information to be able to label all of these on this particular slide. Pause again, please. Go ahead and label everything. then start up, and then we'll talk about what I thought. Okay, so let's go and think about these. So, a taxa or taxon is basically a single species, could potentially be used as a group of species or a gene or set of genes. It's whatever you're putting on the tips of your trees, okay? So taxon is sort of a generic term for whatever you're classifying, all right? And that's okay. Now, a pair of sister taxa, those are two individuals that are closely related, and sister means that they're branching off together, all right? And so the sister taxa here could be Mimi and the taxa next to her. As you can see, it's shown in the slide, but there are other sister taxa here as well. Okay. Now, a monophyletic group. Mono means it has one common ancestor. So any species that are coming off branching from one common ancestor, that's a monophyletic group. Just think of mono as being one. All right. MRCA means most recent common ancestor. Write that down. Most recent common ancestor. Okay, and you can see here the most recent common ancestor is all the way to the bottom of the tree, and that should make sense because it's asking for all the six species. A branch labeled. A node is where you have a speciation event. Okay, hopefully all of this makes sense. Again, I'm just wanting to apply these ideas and to be able to see a tree and then read the tree. Okay, and be able to tell who's related to who's closer related, who's further related, and all sorts of other good stuff. Now, let's talk about a useful group that's very often used in putting together phylogenetic trees, the outgroup. Okay, so an outgroup is sort of like that crazy aunt that no one ever wants to admit they're related to, or that crazy uncle. One. Okay. So what it is that they're distantly related to the group that you're trying to put your tree together on. Okay. And what they do is they're not directly related to the critters in your tree. I mean, they are, but they're not a close relative, so they're distantly related. And because of that, it lets you determine like what traits are considered ancestral and what ones are considered to be shared and derived traits. So to put together our trees, okay, and put together who's related to who, we use what are called synapomorphies or shared derived traits. SYNAPOMOR, synapomorphy. SYNAPOMORPHY, synapomorphy. Okay, and so those are shared derived traits, and you use them to put together who is related to who. Whoever has the most shared derived traits, those are the ones that are the closest related. Okay, now you use the outgroup to determine what traits are ancestral and what ones are derived. And I know that seems a little bit abstract right now, but the more we use this and more we practice it, I promise it will make more sense. So just hang in there for right now. So the reality is we are the products of billions of years of evolution on this planet. And all species have ancestral and derived traits. Okay? We have ancient genes controlling biochemical pathways like DNA replication that are found in all critters because they were inherited from a common ancestor. Those are the ancestral traits. However, some species will have new traits that help them do better in certain environments, and those account for the differences among critters, and those are the derived traits, okay? The synapomorphies, the shared derived traits. So, evolution's complicated, but it explains a lot of things about why we are the way we are, and that's what makes it so cool. Now, it also depends on where you stand in a tree as far as something is ancestral or derived, okay? For example, if you think of evolution before eukaryotes were even around, okay, the nucleus, 3.5 billion years ago, was actually a newly derived trait for all eukaryotes, okay? But if you're looking at humans, well, that's actually an ancestral trait, if that makes sense, okay? So again, you're comparing all the living critters on the planet. Well, we've had a nucleus for 3.5 billion years, and that in some of the earliest cells is what makes a eukaryote. Think about what we've talked about before. So it's where you're standing on the tree as far as if something is ancestral or derived. That's all this is meant to illustrate. So don't let this confuse you, okay? Keep it simple. Okay, so if we're considering this and we're learning how to read a tree, okay, so a shared derived trait would be trait X. Then what I want you to do is think about, okay, so then who should have trait X? Should it be A only, B only, C only, D, or E? What's the correct answer? Hopefully this is easy and you know it's B and C. Okay, so the answer would be E, and that should make sense because you use the traits to illustrate what differentiates them from, in this particular example, A. All right, so B and C are the ones that have trait X. Oh, look, another practice question. Okay, so it's asking what's a shared derived trait for oaks and yews. All right, so you want to find the traits that only oaks and yews have, but that both of them have. So take a moment, read through this, and choose your answer. Okay, so hopefully D makes sense. Tree, the tree form, so like looking like a tree. Okay, and then the production of seeds. That's what oaks and yews have in common. So true leaves you might have chosen as one of the potential choices, but remember, it's just asking only oaks and yews. And when it comes to true leaves, the other species have it there as well. So you wouldn't, ferns have it too, and so you wouldn't want to choose that one. It's all in how the question is worded, okay? And only focus on what it's asking for. Okay, so given what we've talked about, stop the slide, okay, answer the question, and synapomorphy, homology, you can think of as being similar. Homology means, remember, a common ancestor had it, so the offspring had it. But, that's homologous. Don't let that trick you, okay? So, yeah, so I'm going to tell you right now, the first one's a little bit tricky, because the common ancestor had it, but then other individuals had it, but that you could also, depends on where you're standing in the tree, okay? So flip to the slide in a second after you've answered the second one. The second one's much more straightforward. Now, this first question, obviously, I don't like. It's a little bit dicey, and the reason is because the common ancestor had it, and then other individuals have it. However, you could have a species that would lose it, in which case then it would make it hard for you to put it together, okay? So especially if you have distantly related species. So what's the most helpful for constructing trees? Shared derived traits, synapomorphies, as I said, okay? Keep it simple. And then with regards to the shared derived trait for beetles and butterflies, pupa. Okay, that's what puts those two together. And so those are the traits you use to actually put together your tree. Okay, so looking at this particular information, okay, you're given this tree where you've got eukaryotes, which remember means true nucleus, and then you have bacteria, which you should know as far as the nucleus where that stands. So where on this tree would you mark the trait of the nucleus? Take a moment and do that. Then they rearrange the tree down below. Okay, and so what they're wanting you to do is figure out which of the trees below is false if you're given the larger tree above. The first thing I don't want you to do is panic when you see a question like this, okay? Because of the fact that all you have to do is look at the relationships and the answer is there. You just have to take the time to piece it together. Okay, so given this information, it should make sense that the nucleus is placed at the base right there. Because we know bacteria do not have a nucleus. That's what makes them in their prokaryote group. And the eukaryotes, true nucleus, they do. Now, given the information which one is false, you should easily be able to tell by looking through these that D is false, okay? Because of the relationship between the yeast, the lilies, and the pines. You know that the lilies and pines, if you look at the tree above, are the closest related. So those are the sister taxa up there, so they should be the sister taxa down there, which is why D is not, D is definitely false. Now, there is a recent push to start treating people with medicine and sort of considering some evolutionary information, okay? And one way you can do this is to figure out where a disease evolved and who was patient zero. And when you figure that out, then you start realizing how diseases are spread. And if any of you guys are interested in public health, by the way, which, good Lord, talk about being useful right now. All right, the difference between public health and being a doctor as far as medicine is concerned, you can be a doctor in public health, of course, as well, is that in public health, you're trying to figure out where a disease started and how to contain it. Gee, is that useful right now? Whereas, being a doctor, also useful, don't get me wrong, but then you're trying to treat individuals, okay? So if you actually take, and they're doing this now, by the way, with COVID, they're sequencing it and trying to figure out where the disease started, okay? Because that's useful, because if you know who patient zero is, where it happened to start, then you can figure out how it spreads, and then you start figuring out the mechanisms of how it spreads. And if you're interested in this type of information, which I got to tell you, personally, I think this is fascinating, then it's totally an area to go into. Now, another way that this is useful, and this is a perfect example when I argue with my daughter, because I don't know how many times I've, not that I argue with my daughter a lot, But she told me one time, well, what use is, this when it comes to medicine? And that was like a knife through the heart, I gotta tell you. And I don't know if any of you are interested in forensic science or if you ever watch the ID channel. My husband and I jokingly call that the murder channel. By the way, don't watch that too much. You start getting paranoid. However, you can actually use the strains of the genetic strains of a disease, and it has been used in the past in a murder trial, believe it or not. Okay, so there was a particular case when a nurse was having an affair with a doctor. Okay, and strangely, she ended up getting a case of HIV and hepatitis C. Now, this is a horrible case, keep in mind, okay? But she had claimed that she threatened to break off her relationship with this doctor, but yet she continued to see him for vitamin shots. Guys, if you break off an affair with someone, don't go back to them for vitamin shots or, any service whatsoever, okay? I don't know what she was thinking, I gotta tell you. But after this particular treatment, she came up with both of these diseases, by the way. Okay, so how in the world would you solve for this, and how in the world would you find evidence for it? Well, the prosecutors were a clever bunch. Okay, and so what they started to do was they started to get samples of HIV from the doctor's patients. Okay, as well as hepatitis. And then they used a phylogenetic tree, believe it or not, to figure out the relationship of her diseases versus his patient's diseases. Okay, and seriously, how cool is that? I also watch Too Much ID channel. I don't know if you guys do too, but I love the fact that you can actually do this, and it's absolutely fascinating. And go ahead and flip the slide because the data is so cool. So using a phylogenetic analysis of her two diseases as well as the patient's diseases that he treated, you will see that her diseases came straight from a couple of his patients. Okay, so what this means is that he did try to murder her by injecting her with HIV. It's the only way she could have got it, when he had the vitamin shot. And by the way, he's still serving. The doctor is serving a 50-year prison sentence. All right, so again, phylogenetic analysis is used for so many amazing different things. Another way you can actually use this, by the way, and this is incredibly important, is to figure out what meat is actually being used in fish markets. Okay, so we know that there's a shortage of certain types of fish that there's a demand for. And it turns out that people are actually harvesting some species that are not legal and then marketing them as those that are legal. Okay, so one thing I'm going to try to remember, I need to remember to do this. I don't know if you guys have time, but there was a biology teacher who actually asked her students, okay, so if you eat sushi, how do you think the species that you have is what you actually are supposed to have? And so the students were asked to save a sample of their sushi, okay, and she actually sequenced the sushi and looked to see what species they were. And if you guys look this up on Google, I think you'll be able to find it. There's a lot of situations where things are not what they are supposed to be. Okay, so take a moment now. It's totally worth your time. And if I can remember, I'll put it up at the end of lecture in our class too. Google, sushi sequencing biology teacher. She's a high school biology teacher. And by the way, this experiment was brilliant. When she thought of this, it was amazing. And I think she was on Instagram. She was posting her results, but it's all posted on Google. But check that out. It's another way that, using phylogeny to regulate, what species are actually being harvested and what ones aren't. So, and it's becoming more and more important with time because, you don't want to harvest the illegal ones because then it's just going to kill them off. And, then they'll become extinct, and that's no good for anybody. Now, there are situations, by the way, that you can't always resolve evolutionary relationships with the data you have, and that's called a polytomy. Okay, so a polytomy is when you have 3 or more species and you don't know what the relationships are, so you kind of put them, they look like a comb, okay, in the tree. And so look at A, B, and C and figure out which one of these, are we lacking information on where we can't resolve the relationships. What to look for, again, is wherever you see a comb, okay? And C looks like a comb, which means C is a polytomy. It'd be perfect, by the way, to have you guys label that if you are putting a tree on the upcoming exam and ask you guys to identify things. Okay, so when you're building a phylogenetic tree, these are the rules, okay? So you choose informative, homologous, / shared, derived, or synapomorphic traits. You choose an appropriate outgroup. So you want it to be a distantly related aunt or uncle, not too distantly related, okay? But you don't want it to be in the groups that you're trying to classify. You compare the character states of all the taxa, so you're comparing all these traits among them. You identify shared derived characters, okay, again, synapomorphies. You compare all the possible trees that can fit into this matrix, and to be perfectly honest with you, in this day and age when we do this, we use a computer program. And a lot of times if you have, for example, 50 species, look at all the different possible trees you have. That's a nightmare. So you enter the parameters into your computer and tell it, what it should be looking for. And by doing so, then it picks the strongest tree based on the data that you've entered. So let's say you have a couple of different trees that are possible given the information. Okay, and this happens, by the way, all the time. We use what's called parsimony. So parsimony, you pick the most parsimonious tree. And basically what that means is the simplest one that would have evolved. Okay, so if you have a choice between a really complex tree, for example, number 2, or a more straightforward tree, for example, number 1, you're going to pick number 1. And so the reason is that we would not choose number 2 is that if number 2 were true, you'd have to have certain traits basically disappear or de-evolve. And there's a lot more steps in order for number 2 to happen. Always pick the more straightforward, the one where you have, the less steps when you're choosing your tree. So the easiest way for the tree to have happened, okay? So in this situation, don't pick 2. Another way you can kind of think of this is a more straightforward tree. Okay, so of the 2 trees, number 1 is much more straightforward than number 2, and you would have less evolutionary steps. Makes it the less evolutionary steps, then the more parsimonious the tree, and the more likely it is to be the representative of what happened in the past. Okay, so now what I wanted to do is actually practice this. Okay, and so what we're going to do is use DNA sequencing, which, by the way, is used very commonly to put trees together today. So consider this a useful exercise. Of course, all of the ones we give you are useful, but we do this in real life. So what you're going to do is you're going to indicate the location of changes on the trees. Okay, so you have, first off, look at the table at the very bottom right. Okay, those are the sequence. That's the sequence data for your common ancestor, which is at the very bottom, and then the 4 different species, which are all pictured in that purple box towards the top. So that's species 1, 2, 3, and 4. Okay, then trees, you have 3 choices for trees. Tree A, which is to the left, B in the middle, and C to the right. Okay, what I want you to do is you're going to count the number of changes on each tree. Okay, and you're going to draw hash marks to show how many mutations have happened for each species from the common ancestor given the tree. So let's go through this and work through this together, okay? And let's start with tree A. So tree A is putting species 1 and 2 as sister taxa. And by the way, that phrase should make sense. It's also putting 3 and 4 as sister taxa, okay? And you can see the common ancestor towards the very bottom. All right, now what you're looking for is, given this relationship, how many mutations are found, between each of these, okay? So let's start with species 1. So compare species 1, which is at the top of your table, down to the common ancestor, which is at the bottom. Okay, you will see that species 1 has got 2 mutations. One is at site 1 and one is at site 7. That should make sense. Then what I want you to do is draw 2 hash marks right underneath the number 1 on tree A. Okay, then what I want you to do is go to species 2 and count, okay, according to species 2, compare that to the common ancestor. How many mutations do you have? Well, you've only got 1. So then you put 1 hash mark under species 2 on your tree, under that number 2. Okay, then you do the same for 3, then you do the same for 4. Now, be aware though, okay, for example, 3 and 4 share some mutations. So for example, share a mutation at site 5 and 6. So those 2 you would actually put down below the common ancestor for 3 and 4. Okay, and so you would put the mutation for those 2 sites down there, but there are also mutations that differentiate 3 from 4, and you would put those, hash marks up there as well. So stop for a moment, think through this, write down all the hash marks of where the mutations have happened, given these 3 trees, and then say to yourself, okay, which one has the least number of steps, therefore is the most parsimonious and is the most likely tree. All right, then when you're ready, start up lecture again and flip to the next slide. Okay, so hopefully this made sense as I was trying to describe where you're going to put the hash marks and so forth, okay? And so then what you do once you've got your hash marks in there and you're figuring out what mutations have happened, you look for the one that has the least number of hash marks because that's the most parsimonious tree. And if you do this, you will see that tree A seems to be the most parsimonious. Now, go through and match these up if it didn't make sense before. Then what I want you to do is flip to the previous slide, count it up independently and make sure this makes sense, okay? And if it doesn't, let me know. Okay, so what you want to do with a question like this, okay, is you're going to interpret gains and losses on trees, because there are some situations where critters have gain traits and others where they definitely have loss traits. And this is going to happen, particularly if you're looking at individuals or species that are very distantly related, okay? So we see a tree like this. All right, I want you to read the ancestral traits, and then I want you to follow like a road map up to sea lions to see what sea lions have. Okay, so take a moment, read through this, take your time, and choose what you think is the correct answer. So given this information, hopefully D makes sense, okay? Because again, you're going to move up like a road map, over and up, follow the red, okay, up to the sea lions, and that tells you what they have. And whatever is not a derived trait, then they have the ancestral trait, and that's a safe assumption to make. Because what they're showing you at each step of the way, those hash marks, that's what's derived. So if something is not newly derived, then you revert to the ancestral trait. Now, back in the day before sequencing was done, molecular data was collected, they used to use what was called comparative anatomy to put together their trees. So they were just comparing traits between species and seeing who had what and putting their trees together based on that. Well, I can tell you when molecular data came out, everybody got really excited because they thought they would finally answer everything and it would be super straightforward and it would solve all the controversies. Well, yes and no. So there are similarities, okay, and in this particular tree there's a lot of similarities, but there's also some differences. So read through this and on your tree circle where the differences are, okay, and see what you can see as similar and see what you can see as different. By the way, if you're ever asked, hint, hint, on an upcoming exam, hint, hint, to compare and contrast, that means you list how things are similar or you discuss how they're similar, but you also discuss how they're different, okay? That's how you compare them, by saying what is alike, and contrast, meaning you say what is different, okay? Now, I'm not going to ask you a question like this on your exam, but again, I'm trying to prep you for what's in the future. And so they're really big for some reason on rotating trees and then showing that you still have the same relationships. And yeah, I get that that's important, but I'm not that big on it. I think that you should be able to trace who's related to who and realize that you can rotate things around, okay, and that the trees then technically are all the same, okay? So what they're wanting you to do with this question is if you rotate around the node like tree 2 shows, draw what the third tree is going to be, and then also realize that there's an equal sign, so it means the same stupid tree. You just, draw it as they're suggesting it, and I'll show you the answer on the next slide. Okay, hopefully you got something that looks like this, okay? Again, just be aware that that's a big thing. And again, also know that it's all the same tree, okay? All you're doing is flipping the nodes, okay? It just means you're still rotating them around, but the relationships are still the same, and they're still there, and there's really no difference. Okay, so the first thing they want you to do with this tree is to indicate which of these traits is ancestral and which is derived. So as you can see, the blue is a derived trait, okay, and only group C has it. And then the circle is a derived trait, and only B and D have it. Okay, so there's been 2 changes since the common ancestor. So that's easy. Then they want you to identify taxon D is more closely related to A than to B. Is that the case? Well, no, because D shares a trait with B, and those two are sister taxon. You can't really differentiate them anymore based on the characters that you're using. So that would definitely be false. All right, last but not least, this is another application question, okay? So given the 3 trees that you see, they want you to determine which one is correct, so they want you to map the traits. So just like you did with the sequence data, you're going to map how many mutations have happened. You can do that by hash marks, however you want, and then you have to figure out which tree is the most parsimonious, okay? So stop this for a moment. Take the time to do this, guys. I don't ask you to do things like this for no reason. Hint, hint. Okay. So take the time to do this and make sure you understand it. So pause the lecture right now. Okay. And then when you're ready, start it up again. Okay. So with any luck, you chose the second tree. Okay, because there's only 2 changes, whereas the other trees require 3 changes, okay? This ends the material for the first exam, all right? And so what I want you to do the rest of the time, for lecture today is to definitely study. Don't forget, I will be on Zoom on Friday, which, yeah, so by the time you guys are watching this, we'll be on Zoom. And so if you guys have questions, definitely let me know. I look forward to chatting with you soon. Have a great weekend. Stay safe and stay healthy.